So for our project, we chose to focus on the galls of valley oak trees. Our group members are Aleyna Celebi, Dilasha Shenaz, and Nisa Celebi.
What do galls look like? Galls appear on the underside of leaves or twigs of leaves. As you can see, galls can come in many colors, including red, yellow, green, and brown. They can also come in a wide variety of morphological shapes and sizes. Some may look like spherical balls and lumps. You might even see a bunch of galls clustered together. On oak trees, galls are more spherical and can get big as golf balls. Here is a picture of an empty gall. As you can see, in the center of the gall is a larval chamber surrounded by nutritive tissue. Galls can serve as a home and a place to feed and lay eggs for insects. Galls can have one or more larval chambers lined with nutritive tissue and a thin shell of plant tissue that provides mechanical stiffness and strength. The nutritive tissue consists of large, thin-walled cells whose chromosome structure, protein content, and physiology are similar to seed tissue. The larval chambers are morphologically similar in all centipede galls, and specific gall structures result from variation in the development of the outer parenchyma and epidermis. The complexity of these outer tissues varies enormously across gall wasp tribes, species, and between generations. Many people may look at galls on plants and mistakenly think that they are fruits, but they're actually tumors. Galls are abnormal plant growth that can be caused by insects, mites, nematodes, fungi, bacteria, and viruses. In valley oak trees, when a female oak gall wasp, called a centipede, inserts her eggs into the flesh of the plant, the larvae will cause galls to form.
Next, we will focus on types of oak galls. Many types of oak galls can form on oak trees. To the image on the left, for example, we see leaf galls. They are formed on leaf blades or petioles. They are most common galls found on old plants. They appear as leaf curls, blisters, nipples, araniums, which are hairy felt-like growths, and they can be found on the upper or lower leaf surface. And then in the middle image, we can see bud galls. They're also known as flower galls. They are deformed in size and are in the shape of buds and flowers. To the right image, we can see stem and twig galls. They have deformed growth on the stems and twigs, which is what it sounds like, and they range from slight swelling to large knot-like growths. And this type of gall, they appear to be like apples in appearance, and this is a type of gall that's most common in valley oak trees, and they're the twig galls. And this is a type of gall formation that we will be focusing on throughout the rest of our presentation. On to Nisa next. Thank you, Aleyna. Okay, so what causes galls to form? Well, it can be due to irritation and or stimulation of plant cells due to feeding or egg laying by insects. Galls usually form during the accelerated growth period of new leaves, shoots, and flowers in late spring. Insects or mites damage plants by chewing on them. Their salivary secretions, their spit, causes plants to increase the production of normal plant growth hormones. Higher hormone production results in increased cell size or cell numbers. These abnormal cell growths are called galls. Mature plant tissues are usually not affected by gall-inducing organisms. The gall keeps growing as the gall-making insect feeds and grows inside of the gall. And if the gall starts to form, they continue to form even after the insects die. The twigs of valley oak trees that result in apple-like growths are the result of gall wasps. Another name for these are cynipids, and plural it's Snipidae. The snipid gall inducer wasps induce galls of 5 to 20 cubic centimeters on the twigs of valley oaks, an endemic tree in California. Gall growth occurs twice annually during late spring and midsummer. Next, we have developmental stages of gall formation. Synnipid gall development can be divided into 4 phases: initiation, growth, maturation, and dehiscence. First, we have the initiation phase. The female synnipid will find a host plant and determine a location. Then she will oviposition, which means to insert her eggs into the flesh of the plant. As far as is known, gall induction results from secretions derived from the egg and larva, and not from many maternal secretions. Plant cells neighboring the egg lyse, which means rupture, the cells to produce a small chamber. After hatching, the larva enters this chamber and controls all subsequent tissue differentiation. Each larva develops in its own chamber, and galls induced by a specific gall wasp species are usually neither single-chambered, which is uniocular, or multi-chambered, multilocular. Okay, so if we look at our photo in initiation phase of number 1, we can see our little synnipid, and she'll be laying her eggs right on the node of that stem. In the bottom left photo, we can see the gall growth phase takes place while the gall wasp larva remains very small. The larval chamber forms and enlarges a network of vascular bundles that develops and joins those supplying the host organ, and layers of outer parenchyma developed around the larval chamber. Nutritive tissue is the gall wasp's sole source of food throughout development. During the growth phase, the gall acts as a major sink for mineral nutrients and photoassimilates, which is pretty much carbon fixed during photosynthesis. The outer gall tissues synthesize tannins and phenolics, which were originally thought to be a deterrent against free-feeding insect larvae. However, these compounds are now known to be feeding stimulants for some insect species, and an additional function as a fungicide has been proposed. Galls on leaves now grow by increasing the rate of photosynthesis on the affected leaf, and by taking the energy and photoassimilate produced in order to form the gall. Galls also extract energy and nutrients from neighboring leaves and regions of the plant. Next, in the top right, we have the maturation phase. Over here, we can see that the maturation phase is characterized by decreased rates of cell division, and the gall ceases to be a major sink for host plant resources. The larva secretes enzymes that convert plant starch into soluble sugar, which the larva will feed on in the nutritive cells lining the larval chamber. The feeding action triggers a conversion of neighboring vacuolate parenchyma into further nutritive cells. Feeding continues until the sclerenchymatous shell of the larval chamber is reached. And I'm not too sure if you can see, but right here we have our galls. From the right, we have the young before it's mature, and the left we have after it has matured and browned. And next, we have our dehiscence phase. That's the last phase. This is the opening and dehiscence of galls towards the end of the maturation stage, and it represents a period of greatest and the largest amount of physiological change in the tissues that comprise it. Many oak gall tissues surrounding the larval chamber become lignified. This is when the cell hardens on the outside, and in some cases, the regional tissue death occurs in the internal air spaces. The timing of lignification is under the larva's control. Lignification makes the tissue unusable and inedible for many herbivores protecting the larva. Larva also causes lignification of the gall to occur and make the gall drop slightly before leaves fall during autumn, ensuring a covering of leaves and a suitable microenvironment so that they can pupate or enter diapause. And this is just a period of suspended development. And next, we actually have a commercial break brought to you by our sponsors. Thank you, Nisa. Wow, that was truly beautiful.
You're
and
welcome.
a free gall included? What a steal! Next, we'll be focusing on the mechanism behind gall formation. We'll be looking at the genetic aspects and the hormonal aspects. So, comparative transcriptome analysis was done using 4 different insect galls generated on leaves. And comparison of these different galls revealed that photosynthetic genes are downregulated in galls, which supported the previous hypothesis that galls are converted to sink organs rather than source organs on the leaves. And in recent development, they also discovered how developmental cell cycle and phytohormone genes are upregulated in galls. And there's approximately 40 genes that are commonly upregulated in these 4 galls. And in particular, several key regulators of stem cell generation included CLE44, BAM3, WOXes, and maintenance floral organ development of like Sepalata, Agamas, and Apetala1 were found to be commonly upregulated, but the mechanisms of synapid gall induction still remain little known. The larva hijack of chemical signaling pathways by secreting 2 plant hormones, auxin and cytokinin, which induce abnormal cell divisions leading to gall formation. However, the mode of action of these chemical substances and the general mechanism by which the insect could control and manipulate plant development is still not known. And other oak trees form galls. Many other oaks, including red oaks, black oaks, can get infected by synapid gall wasps, but valley oak trees are preferred. What is the biological significance of galls? Gall wasps have a parasitic symbiotic relationship with valley oaks, but in most cases do not excessively harm their host tree. Gall wasps contribute to the diversity of their ecosystem, and they also provide many species with a source of food. Gall wasps are naturally killed by a complex of fungi, parasites, predators, and competing insects, primarily which are moth larvae and other wasps that live within galls. Gall wasps are also preyed upon by various small insectivorous birds like woodpeckers and small mammals. Hello, for our project, we chose to focus on the galls of valley oak trees. Our group members are Aleyna Celebi, Dilasha Shenaz, and Nisa Celebi.
What do galls look like? Galls appear